This module is on glycogen metabolism. What is glycogen? Glycogen is the storage form of glucose. Glycogen is the storage form of glucose. Okay, so how is glucose residue attached to glycogen? Glycogen, it has a primer which is called the glycogen, onto which the glucose residues are arranged in a linear fashion along 1,4 linkages, and at the branch points there is alpha 1,6 glycosidic linkages. So in a linear fashion, the glucose residues will be linked together by alpha 1,4 linkages, and in the branch points it will have alpha 1,6 linkages. So this around this glycogen, it's arranged around in 12 concentric layers. Okay, so around 12 to 14 concentric layers, the glucose residues are arranged with branches. So branches are more towards the bases. Okay, so more towards the base of this that is closer to the glycogen molecule. So here so that more amount of glucose residues are arranged in a compact fashion. More amount of glucose residues are arranged in a compact fashion. So if the glucose residues had to be stored in a linear fashion, then it would take a lot of space. Rather than that, since it is arranged in as branches, it can be a more number of glucose residues can be present or can be stored in a compact small area, right? So this is the function of the glycogen. Now where are glycogen molecules synthesized? It is synthesized in two places. One is your liver and the other one is your muscles. Okay, so glycogen is synthesized in liver and muscle. So the glycogen which is synthesized in the liver contributes to blood glucose. So glycogen which is synthesized in the liver contributes to blood glucose, whereas in muscle it does not contribute to blood glucose. It only muscle utilizes the glucose residues obtained from the glycogen for muscle contraction, for energy purposes. Okay, but since this actually per gram of liver the glycogen content is more. Okay, per gram of liver the glycogen content is more compared to that of muscle, but since the muscle mass is more, we say that more glycogen molecules are present in the muscle. Okay, so but actually per gram of liver contains more concentrated number of glycogen molecules than the muscle glycogen. So this is about the glycogen. So let's just see an overview of the glycogen metabolism. So glucose can be converted to glucose 6-phosphate. This is the first step of glycolysis, right? It is the first step of glycolysis. After this glucose 6-phosphate, it has multiple fates. One of the fate is glucose 6-phosphate can be converted to glucose 1-phosphate by a phosphoglucomutase reaction. The phosphate group is alone changes its position by the enzyme phosphoglucomutase. Now this glucose 1-phosphate can get connected together by glycogen synthase forming glycogen. Okay, so this is the glycogen synthesis reaction. Whereas glycogenolysis is nothing but, what is glycogenolysis? From the glycogen molecules, glucose residues are removed as glucose 1-phosphate by the enzyme glycogen phosphorylase, and this glucose 1-phosphate can be converted to glucose 6-phosphate by phosphoglucomutase. This glucose 6-phosphate can be converted to glucose and it can be released into the blood circulation if it is in liver. Okay, if it is in liver. At the same time, glucose 6-phosphate is converted to glucose by an enzyme called glucose 6-phosphatase in liver, because liver has glucose 6-phosphatase enzyme. So glucose 6-phosphate is converted to glucose in the liver and it is released into the circulation whenever glucose is necessary. But in muscle, glucose 6-phosphatase is not present. So in muscle, it stays as glucose 6-phosphate only, and it enters into further reactions of glycolysis in muscle. In muscle glycolysis, and it serves as a source of energy. Okay, it utilizes the glucose from the glycogen for energy purposes. Okay, so this is the overall view of the glycogen metabolism. So glucose to glucose 6-phosphate, the glucose 6-phosphate can enter into glycogen synthesis, and the rate limiting enzyme is glycogen synthase, right? At the same time, glycogenolysis, the rate limiting enzyme is glycogen phosphorylase. Here the glycogen is converted to glucose 1-phosphate and it is converted to glucose 6-phosphate. In the liver has glucose 6-phosphatase enzyme. So glucose 6-phosphate is converted to glucose in liver and it is released into the circulation. It contributes to blood glucose. But in muscle, glucose 6-phosphatase enzyme is not present. So glucose 6-phosphate enters into further reactions of glycolysis, and it serves as an energy source and is utilized in the muscle contraction. Okay, so this is the overall view of the glycogen metabolism. Now let's get into the steps of glycogen metabolism. First we'll see glycogen synthesis. So if you see glycogen synthesis, the first step is activation of glucose. Okay, glucose, just like that, it is not added to the glycogen. Instead, what happens, it is first activated. So how do you activate the glucose? We've already seen glucose to glucose 6-phosphate. Glucose 6-phosphate gets converted to glucose 1-phosphate. Now this glucose 1-phosphate combines with a UTP, okay, uridine triphosphate, and forms what? UDP glucose. Okay, it forms UDP glucose plus PPI is released. Okay, so here what is the enzyme? UDP glucose pyrophosphorylase. UDP glucose pyrophosphorylase. So which is the active form of glucose? The active form of glucose is the UDP glucose. The active form of glucose is UDP glucose. It is in this form that it is attached to the glycogen. Okay, so here the next step is the glycogen synthase reaction. In the glycogen synthase reaction, what happens? I have told you all of the glucose residues will be attached to the glycogenin, which is nothing but the glycogen primer. Okay, so you need a primer for the formation of glycogen. So into this glycogen primer, all of your glucose residues, that is the activated UDP glucose, will be attached. Yes, UDP glucose will be attached. So it will be attached in linear fashion. In the linear fashion, what is the linkage? It is the alpha 1 4 linkages, glycosidic linkages. Okay, so these linkages are formed between the glucose residues. Now here, along the glycogen primer, when the UDP glucose is added, what happens? The glucose residues gets linked together and it releases the UDP. Okay, it releases the UDP. So here, now linear fashion, the glycogen synthase enzyme can attach these glucose residues in the linear fashion, forming alpha 1,4 linkages. So glycogen synthase can attach glucose residues to the glycogen primer, forming alpha 1,4 linkages. Now, but this is not the only linkage present in the glycogen, right? So we need to form branches so that more number of glucose residues can be stored in the glycogen. So for this we need a branching enzyme. So this branching enzyme, what does it do? Branching enzyme is also called alpha 1,4 to alpha 1,6 glucan transferase, alpha 1,4 to alpha 1,6 transglucosidase. Okay, transglucosidase. This is the branching enzyme. So what does this branching enzyme do? So look at this. What does the branching enzyme do here? The branching enzyme, after initially what happens, the glucose residues are added one after the other. So as the glucose residues are added in a linear fashion, when about 11 to 12 glucose residues are being added in a linear fashion to the glycogen primer, the last 6 to 8 residues, okay, the last 6 to 8 residues, what happens, they will be removed from this linear arrangement, and it will form a separate branch with the molecule, okay, separate branch with the another molecule. So in this it will form alpha 1,6 linkage, okay, in this it will form alpha 1,6 linkage. So here a new branch is being created with alpha 1,6 linkage, and who forms this new branch? It is the branching enzyme, which is nothing but the alpha 1,4 to alpha 1,6 transglucosidase enzyme. So here after the branching enzyme forms a branch point, after that there is linear arrangement, linear attachment of glucose residues forming alpha 1,4 linkages. Okay, so alpha 1,6 linkage is only found in the branch point, after which it is linear attachment of the glucose residues with alpha 1,4 linkages. So branch point, what is the linkage? It is alpha 1,6. Linearly, what is the linkage? It is alpha 1,4 linkages. So this is more number of glucose residues are attached, and finally the storage form of glucose, which is called the glycogen, is formed. So where is it formed? It is formed in the liver as well as in the muscle. Okay. So always remember, only liver glycogen contributes to the blood glucose. Muscle glycogen does not contribute to blood glucose. Okay, so it is utilized only by the muscle. So what are the 3 steps involved in the glycogen synthesis? First step is you have to activate the glucose, forming UDP glucose. So the active form of glucose will be the UDP glucose. Now after which UDP glucose, with the help of the enzyme glycogen synthase, it will attach the glucose residues to the glycogen primer, forming alpha 1,4 glycosidic linkages. Now UDP is released in this reaction. Now after which we have to form branch points. So the branching enzyme comes. The branching enzyme, after 11 to 12 glucose residues are being attached in a linear fashion, the last 6 to 8 residues are being removed from the chain, and it will form a branch point. So in the branch point, the linkage is alpha 1,6 linkage, after which the glucose residues are attached in linear fashion with alpha 1,4 linkage. So alpha 1,4 linkage is always formed by glycogen synthase, and alpha 1,6 linkage is formed by the branching enzyme. So finally more number of glucose residues are being formed, and the storage form of glucose, which is the glycogen, is formed. So which hormone stimulates glycogen synthesis? So whenever you have more number of glucose in the circulation, you tend to utilize the glucose for energy, and rest of the glucose you tend to store it in the form of glycogen. So which hormone is necessary for this? It is insulin. So insulin stimulates glycogen synthesis. Insulin stimulates glycogen synthesis or glycogenesis. Okay, insulin stimulates, whereas glucagon, epinephrine and all will decrease the glycogen synthesis. So this is about glycogen synthesis. Now let's talk about glycogenolysis. Now let's talk about glycogenolysis. So what is glycogenolysis? Glycogenolysis is the breakdown of glycogen. Yes, glycogenolysis is the breakdown of glycogen. So what is the first enzyme involved here? It is the glycogen phosphorylase. So phosphorylysis is the first step here. Okay, glycogen phosphorylase is the enzyme. Phosphorylysis is the first step here. So what happens in phosphorylysis? From the glycogen, so look at this is a glycogen molecule. Here we have linear arrangement, we have glycogen, we have branch points to which again glucose residues are attached, right? So this is the glycogen molecule. So from the glycogen molecule we have to remove the glucose residues one by one. So how much ever glucose you are in need of, that much of a glucose residues will be removed. So whenever you're in fasting state, so 5 hours after meal, okay, that is a after post absorptive state, your body will be having reduced amount of glucose. So what happens, glycogenolysis from the liver happens and it will reduce glucose, it will give off glucose to the blood. Yes, so that process is called the glycogenolysis. So here, how do you remove this glucose residues? First you have to break this alpha 1,4 linkage. Alpha 1,4 linkage is formed between the between the glucose residues which are attached in a linear fashion. So alpha 1,4 linkages are broken down. So what is the function of this glycogen phosphorylase? It breaks down the alpha 1,4 linkages. It contains pyridoxal PLP, or the pyridoxal phosphate acids prosthetic group. Understood? Now here, since one alpha 1,4 linkages are being broken down, and what is being removed? The glucose is removed as, how is it removed? The glucose is removed as glucose 1-phosphate. Okay, so we'll come to that. Now here, after that, when it comes, when all of these glucose residues, are being removed, okay, now we have this branch point again, right? We have this branch point. So branch point, along the branch point, how will it get broken down? So some about 3 to 4 glucose residues before the branch point, the glycogen phosphorylase action will stop, and the second enzyme, which is the bifunctional enzyme, okay, bifunctional enzyme which has two enzymatic activity. The first enzymatic activity is nothing but alpha 1,4 glucan transferase, okay, alpha 1,4 glucan transferase. So here what happens, the three of this last glucose residues will be removed from the branch point, and it will be transferred to the another chain, okay, it will be transferred to the another linear chain, okay, forming alpha 1,4 linkage here. Now we have only one glucose residue having alpha 1,6 linkage. Now this alpha 1,6 linkage is broken down by the second enzyme of the bifunctional enzyme, debranching enzyme, which is nothing but our alpha 1,6 glucosidase. Alpha 1,6 glucosidase. Now this alpha 1,6 glucosidase will break this alpha 1,6 linkage here, and it will liberate this as free glucose. It will liberate this as free glucose. So how are these glucose, how are the other glucose residues removed? The other glucose residues are removed as glucose 1-phosphate. Okay, other glucose residues from the glycogen phosphorylase activity are removed as glucose 1-phosphate, but after the alpha 1,6 glucosidase activity from the branch point, alpha 1,6 branch point, that one glucose residue is alone removed as free glucose. Yes, do you understand? Now what happens to this glucose 1-phosphate which is being removed? So from the glycogen, glucose 1-phosphate is only removed one by one, right? So what happens to this glucose 1-phosphate? Now this glucose 1-phosphate, we have seen already, glucose 1-phosphate undergoes a phosphoglucomutase reaction. Phosphoglucomutase reaction, and gets converted to glucose 6-phosphate. Glucose 6-phosphate. Now you know glucose 6-phosphate can be converted to glucose by an enzyme called glucose 6-phosphatase. Okay, by an enzyme called glucose 6-phosphatase. Now this glucose 6-phosphatase is present where? It is present only in the liver. Is it present in the muscle? No, it is not present in the muscle. So what happens in muscle? In the muscle, glucose 6-phosphate enters directly into the glycolysis step. Okay, enters into glycolysis step, and it is used as a source of energy for muscle contraction. Now, since glucose 6 phosphate is the product of the first step of glycolysis, right? So first, whenever in a glycolysis reaction, how many ATPs do we get? Normally, when free glucose enters into glycolysis reaction, how many ATPs do we get? We get 2 ATPs, okay, 2 ATPs. But when directly glucose 6 phosphate in the muscle, what happens? This glucose 6 phosphate enters into the glycolysis reaction, right? So from the glycogen molecule in the muscle glycogen molecule, glucose 1 phosphate is released. This 1 phosphate is converted to 6 phosphate by a mutase reaction. This 6 phosphate now enters into the glycolysis. So the first step, that is the hexokinase step, is bypassed here. Yes, hexokinase step is bypassed here. So what happens? How many ATPs do we get? We get 3 ATPs. So plus 1. Yes, plus 1, because in hexokinase 1 ATP will be utilized. No, so we are bypassing this step in this whenever glucose 1 phosphate is converted to glucose 6 phosphate. Whenever glucose from the muscle glycogen is removed, the hexokinase first step of glycolysis is bypassed, so ATP is not utilized. So one more ATP extra we are getting. So we get 3 ATPs whenever muscle glycogen, glucose from the muscle glycogen enters the glycolysis. Understood? So this is one significance. So here, so in liver what happens, in muscle what happens, you know. So this is the steps, these are the steps of glycogenolysis. Okay, so what is the first reaction? Important reaction is the glycogen phosphorylase. Now this glycogen phosphorylase breaks the alpha 1,4 linkages in the linear arrangement of glucose, and it releases these residues as glucose 1 phosphate. Understood? Now next, along the branch point what happens? Now about 3 to 4 residues away from the branch point, this glycogen phosphorylase action stops, and this alpha 1,4 glucan transferase enzyme will come, and that will remove these last three glucose residues, and it will transfer it to the another chain. It will transfer it to the other chain. Now what happens? One glucose residue with a branch point alone is remaining. So that is being broken down by the alpha 1,6 glucosidase enzyme that breaks the alpha 1,6 linkage, and it liberates that residue as a free glucose. Right? So glycogen phosphorylase liberates the residues as glucose 1 phosphate. Alpha 1,6 glucosidase will liberate the glucose at the branch point as free glucose. So finally, again an unbranched polymer is obtained. From this unbranched polymer, again glycogen phosphorylase will act and remove these glucose residues as glucose 1 phosphate. Now this glucose 1 phosphate gets converted to glucose 6 phosphate, right? And then this glucose 6 phosphate is converted to glucose by an enzyme called glucose 6 phosphatase, which is present in the liver. In muscle, this glucose 6 phosphatase is not present, so it can enter directly into further steps of glycolysis. So since one step is bypassed, that is the first step, hexokinase glucokinase step is bypassed, what happens? One ATP is extra for us. So after, when a muscle glycogen muscle glycogen releases glucose and that enters glycolysis, we get extra 1 ATP than the normal glycolysis which happens from the free glucose. Okay, from the free glucose we get 2 ATPs, but from the glycogen, when the muscle glycogen releases glucose 1 phosphate, it gets converted to glucose 6 phosphate that bypasses the first step of glycolysis, so we get 3 ATPs. Yes, understood? So with this we are completing the glycogen metabolism. Okay, now let's see the glycogen metabolism regulation. So regulation part is very important. Now there are two things which you have to remember. One is the hormonal regulation. Hormonal regulation, and the other one is the covalent modification. So covalent modification is nothing but regulation of the enzymatic activities by either phosphorylation or dephosphorylation of the enzyme. Now let's see hormonal regulation. You have to remember about glucagon and insulin. So insulin favors, it's an anabolic hormone. So insulin favors glycogen synthesis. Yes, glycogen synthesis. So how does insulin act? Insulin activates the enzyme by dephosphorylating it. Okay, dephosphorylating the enzyme. So it dephosphorylates the enzyme and makes the enzyme active. So enzymes of glycogen synthesis will be active in its dephosphorylated state. Yes, understood. Likewise, glucagon, epinephrine, these hormones, what does it do? They favor glycogenolysis. Yes, so these hormones will activate the enzymes of glycogenolysis by phosphorylating them. So enzymes of glycogenolysis are active in the phosphorylated state. Understood? So with this itself you can understand the covalent modification, which is nothing but the phosphorylated and dephosphorylated state of the particular enzyme, which makes it active or inactive. Now let's see when glucagon, how does a glucagon act? Glucagon and epinephrine, these enzymes, how does it act? How does it act? They attach to a cell surface receptor and they activate adenylyl cyclase. Okay, they activate adenylyl cyclase. The adenylyl cyclase converts ATP to cyclic AMP. It converts ATP to cyclic AMP. Now this cyclic AMP will activate protein kinase. Okay, it will activate protein kinase. So protein kinase active form is formed. Now this protein kinase, the action of this protein kinase is to phosphorylate. So what does it phosphorylate? It phosphorylates an enzyme called phosphorylase kinase. Okay, it activates phosphorylase kinase. So phosphorylase kinase is phosphorylated and is active. Okay, it is enzyme is phosphorylated to its active form. Understood? That phosphorylation is done by the protein kinase, active protein kinase. Now this phosphorylase kinase will now phosphorylate glycogen phosphorylase enzyme. Okay, glycogen phosphorylase enzyme. Now this glycogen phosphorylase enzyme is now activated. How is it activated? It is activated by phosphorylation. So phosphorylation of this enzyme happens and glycogen phosphorylase will now become active. Now what is the function of glycogen phosphorylase? Glycogen phosphorylase is an enzyme of glycogenolysis. It's an enzyme of glycogenolysis. Yes, similarly this protein kinase or the phosphorylate kinase can phosphorylate other enzyme like your glycogen synthase. So what did I tell you? Glycogen synthase, if it is phosphorylated, what will happen? It will become inactive. Okay, it will become inactive. So this becomes inactive. So glycogen synthesis becomes inactive whenever glucagon is stimulated. Are you able to understand? So glucagon, what does the glucagon hormone does? It acts by increasing the cyclic AMP. Now cyclic AMP will activate protein kinase. Now the active protein kinase will phosphorylate phosphorylase kinase. Now this phosphorylase kinase will phosphorylate glycogen phosphorylase. So phosphorylation of glycogen phosphorylase makes the enzyme active. So glycogenolysis will take place. Similarly, this protein kinase, it not only phosphorylates the glycogen phosphorylase, it also phosphorylates one more enzyme, which is the glycogen synthase. But phosphorylation of glycogen synthase makes it inactive. So glycogen synthesis is inactive. Okay. So this both glycogen synthesis and glycogenolysis does not happen simultaneously. It is regulated in this kind of a manner. Yes, you are able to understand, right? So this, so you have to know about this regulation. So if you know what does insulin do, what does glucagon do, and the enzymes are active in phosphorylated state or dephosphorylated state, then it will be easy for you. Now this cyclic AMP, how is it regulated? Increased levels of the cyclic AMP only forms all this, right? So it brings about all this phosphorylation and activation of glycogenolysis. Now if I inhibit, if I decrease the levels of cyclic AMP, how can I decrease? By an enzyme called phosphodiesterase, which can metabolize cyclic AMP to AMP. Okay, so thereby what can happen? The levels of cyclic AMP can decrease. The levels of cyclic AMP can decrease. So if I decrease the levels of cyclic AMP, then this phosphorylation process, whatever I had told, will not happen. Yes, so this phosphorylation of glycogen synthesis also will not happen. So dephosphorylation of this glycogen synthesis can be brought about by which enzyme? Which hormone? Insulin. So insulin can bring about dephosphorylation. So dephosphorylation of the glycogen synthase enzyme can activate it and glycogen synthesis can occur. Okay, so insulin activates phosphodiesterase enzyme also. One more enzyme which is protein phosphatase. Okay, one more enzyme which is protein phosphatase 1, that can decrease the level of phosphorylase kinase. Okay, that can bring about dephosphorylation of this phosphorylase, that is, it can remove the phosphate from this phosphorylated enzymes. Okay, so the dephosphorylation of this phosphorylase enzyme can be brought about by protein phosphatase 1, that is also activated by insulin, so the phosphorylation process will not happen. So everything is removed. So insulin brings about dephosphorylation of the glycogen synthase and glycogen synthesis can take place. So this is the regulation of the glycogen metabolism. So what is the key enzyme of glycogenolysis? So glycogenolysis, what is the key enzyme? It is the glycogen phosphorylase. Okay, and what is the key enzyme for glycogen synthesis? The key enzyme for glycogen synthesis will be glycogen synthase. Yes. So this is the rate limiting enzymes that you should know. So also this is about glucagon and insulin. You have to know glucagon activates the enzyme by phosphorylating it. Insulin activates the enzyme by dephosphorylating it. Okay, and how does cyclic AMP levels alter all these phosphorylations and dephosphorylations, that you have to understand. Yes, thank you.